Yes, hello, I'm Delphine Uineza. I'm a student at University of Ines Ruhengeri. I'm doing there biomedical laboratory science in the Faculty of Health Science. So I'm here to talk to you, to tell you about the immunology too, which is applied immunology. If we know immunology, there is a basic immunology, and now we are going to deal with applied immunology. So we are going to deal with two parts. The first one is the laboratory practical ELISA, and the second is going to be the principle of vaccination. So to understand it well, for laboratory practical ELISA, I prepared there is a three questions which are going to help us to understand it very well. The first question is what is the principle of ELISA techniques? This one is going to help us to understand well how this ELISA technique work. The second is going to be how is a sandwich ELISA is different from indirect ELISA. So ELISA is having many different types, sandwich and direct. There is even another, but here we are going to deal with sandwich ELISA and indirect ELISA and how they are different. So the third one is going to be what are the application of ELISA in a clinical practice. After knowing all about ELISA, we're going to know where ELISA is applied. So for the second one, the principle of vaccination, we are going to deal also with the three questions which are going to help us to understand very well vaccination. The first is what are the main types of vaccine and their characteristics. The second is going to be how does health immunity contribute to disease prevention. The third one is going to be what is the difference between active and passive immunization. So let us proceed by starting for the first one, for the ELISA. So as the title said, that is a laboratory practical ELISA, and we are going to see the principle of the ELISA technique. This ELISA technique, first of all, have to know what is ELISA. The ELISA is, in full word, is called enzyme-linked immunosorbent assay, and this is biochemical techniques used to detect and quantify substances such as proteins, antibodies, or hormones. So the principle of this ELISA involves, there are three principles. It works in three principles. The first one is antigen antibody specificity, enzyme substrate reaction, and quantification. For antigen antibody specificity, ELISA relies on the specific interaction between an antigen and its corresponding antibody. The second one, enzyme substrate reaction. An enzyme is conjugated to an antibody or antigen. When a substrate is added, the enzyme catalyzes a color change or another detectable signals. So for the quantification, the intensity of the signal is proportional to the concentration of target analyte, allowing for quantitative analysis. So this is the principle of how ELISA techniques work. It works in three: antigen antibody specificity, enzyme substrate reaction, and quantification. So this is the diagram which explains it very well. The first, specific antibody bound to the bottom of where. We just like to use the where where we perform the techniques of ELISA. The second, sample containing unknown amount of antigen is added to the where, to that specific antibody which was bound to the where, to the bottom of the where. The third, antigen binds first antibody. The antigen you added, it immediately binds to the antibody which was at the bottom of where. The fourth, antibody linked to enzyme is added to detect the antigen. This antibody is likely to be the secondary, because the one which was at the bottom was the primary, and this one is the secondary antibody which is linked to an enzyme. That's why it is called the conjugated antibody. This antibody linked to enzyme is added to detect the antigen. Substrate is also added, and change occurs. Immediately the color change. So we are going to see how a sandwich eraser is different from an indirect eraser. So these are the features: sandwich eraser and indirect eraser. Sandwich eraser measure antigen directly, while indirect eraser measure antibody in the sample. They are different. The sandwich eraser detect antigen, while indirect detect antibody in the sample. The sandwich eraser capture antigen between two antibody, while indirect eraser uses an antigen to detect and quantify specific antibody. This sandwich ELISA is having high specificity, while indirect ELISA is having moderate specificity. Sandwich ELISA also is higher due to two antibody binding sites. Indirect ELISA is amplify the signal via secondary antibody. Sandwich ELISA also detect a cytokine in a patient's blood, while indirect ELISA detect HIV antibody in the blood. So we are going to proceed with the application of ELISA. This ELISA is applied in many clinical practice, such as disease diagnosis, allergy testing, hormonal assay, cancer biomarker, drug testing, vaccine evaluation, food safety, and quality control. Those are application of ELISA techniques in clinical practice. So let's move on our second title, which was a principle of vaccination. So to understand it well, also these questions are going to help us to understand it very well. We are going to deal with the many types of vaccines and their characteristics. Vaccine can be classified into several types based on their components and how they stimulate the immune system. So these are the different types of the vaccine. The first one is inactivated vaccine, live attenuated vaccine, subunity, recombinant polysaccharide or conjugate vaccine, toxoid vaccine, DNA, and messenger RNA vaccine. So starting from the first one, inactivated or killed vaccine. Its characteristic, it is made from viruses or bacteria that have been killed or inactivated so they can no longer replicate. This is inactivated vaccine. The second one is live attenuated vaccine. Its characteristic, it contains live pathogens that have been weakened so they cannot cause a disease in healthy individuals. They contain the live pathogen but these live pathogens have been weakened. They're not acting normally. They become weakened so that they cannot cause a disease in healthy individuals. For subunit vaccine or conjugate vaccine, its characteristic means it uses the parts of the pathogen, not a whole virus or whole bacteria, but some parts of it rather than the whole pathogen. Yes. Toxoid vaccine, its characteristic to contain inactivated toxins produced by bacteria rather than the bacteria themselves. DNA and messenger RNA vaccine, its characteristic, it uses genetic materials to instruct cells to produce antigens that stimulate an immune response. So this is an mRNA vaccines. So we proceed with how does herd immunity contribute to disease prevention? First, you have to know what is herd immunity. Herd immunity occurs when a significant portion of populations become immune to an infectious disease, either through vaccination or previous infection, thereby reducing the overall amount of the pathogens spread. So, the key was herd immunity contribute to disease prevention include the first one, reduced transmission. When enough individuals are immune, the pathogen has fewer hosts to infect, which slows down its spread within the population. Second one, protection of vulnerable individuals. People who cannot be vaccinated, those are vulnerable individuals. Example, infants, elder, immunocompromised individual. These people cannot be vaccinated. Are indirectly detected because the spread of the disease is minimized. The third one, preventing outbreaks. Health immunity can help prevent outbreaks or epidemic of vaccine preventable disease by lowering the overall number of susceptible individuals in the immunity. This is how the health immunity contributes to disease prevention. So proceed with this question also, what is the difference between active and passive immunization? So we are going to see how the active is different from passive immunization. So starting from active immunization, and this is the mechanism of how active immunization works, involves stimulating the immune system to produce its own antibodies in response to an infection or vaccination. Here, the immune system is stimulated so that it produces its own antibody when there is infection or vaccination in the body. So, duration of active immunization. This active immunization is long-lasting or permanent immunity. Means that it lasts in years or even a lifetime. Even people can have it with a whole life, if with her or his whole life. Example: vaccine, natural infection vaccines for polio. Yes, because for the babies who are newborn, they use to vaccinate them for this disease, polio, measles, and so on. So this is the kind of active immunization, the vaccine which take long time within the body of an individual. So plus these are like advantage to provide lasting protection against future infection. While it disadvantage, it may take time for the immune system to mount an effective response, and it requires a vaccination or exposure to the pathogen. Yeah, we continue with the passive immunization, and this is the mechanism of how it works. It involves the transfer of preformed antibody from an immune individual to a non-immune individual. So it's very different from the passive, because for passive it is going to be stimulated because of the infection or vaccine, but here it involves the transfer of a preformed antibody from an immune individual to a non-immune individual. So duration of passive immunity is a short-term immunity. It lasts in weeks or months. It is not like active. Active, it takes years or lifetime of the individual, but here it takes weeks or months, since the body does not produce its own antibody. Here the body does not produce its own antibody. The antibody which it uses is preformed. It is formed outside and then it is injected within the bodies of an individual. Example, administration of monoclonal antibodies. Yes. Example for rabies or COVID-19. Breast milk, it is natural passive immunity also. This does not last in a long time. Immunoglobulin therapy. Its positivity provides immediate protection or treatment. Useful for people who are at high risk of infection. So its disadvantage, its immunity is a temporary, very temporary. It lasts in a short period of time. It can't, people can't be with it in their whole lifetime, as the body does not produce its own immune response. So this is all about the immunization, immune system. So thank you for watching, thank you for listening, and this is very important to know it. So thank you for watching, thank you for being with me.